Let's take a look at aerobic respiration. We're going to use glucose as our model molecule. The overall equation is shown right here. So we talked about that in the previous video also. So we're breaking down glucose as our model molecule into CO2 and water, and we're generating ATP out of that. So of course, you can digest other molecules too, but glucose, that's the model molecule that's usually used for this. Aerobic respiration is a three-part process, and the first part is called glycolysis. In glycolysis, we're going to take glucose, and then we're going to take a bunch of metabolic steps, and we're going to end up with pyruvate, 2 pyruvates that is. So we essentially take a 6-carbon carbohydrate, glucose, and break it down into 2 3-carbon carbohydrates, pyruvate. So essentially you're breaking the glucose in half, and that's it. For each glucose molecule, you're only going to get 2 ATPs out of this process. Grand total is 2. We're going to do the ATP accounting in more detail later, but if there's oxygen available and there are mitochondria available, then the cell will take the pyruvate and convert it into acetyl-CoA, which is a 2-carbon compound. So that means we're going to lose a CO2 here in the process, and we'll talk about that some more later too. The acetyl-CoA, that 2-carbon compound, will enter the Krebs cycle, and in the process it will completely oxidize to CO2. So there's a bunch of CO2 leaving. And for each round of the Krebs cycle is 1 CO2 because it should be 2 ATP. So anyway, I don't think they're counting here right, but for each acetyl-CoA, the 2 carbons they're leaving in the Krebs cycle as 2 CO2s. We will also gain a grand total of 1 ATP per round of the Krebs cycle. So since 2 acetyl-CoA are entering here, we're going to end up with 2 ATP. But again, we're going to do the accounting of all these numbers a little bit later. And then once we have completely oxidized the glucose, or the original glucose molecule, then we can use these hydrogens that we took off in the process of oxidation. We can move them on to NADH and NADPH, those coenzymes, and they will then move these electrons and hydrogens over to the electron transport chain, where we then have major ATP output. So this is your overview slide. I admit it's a little bit crowded, but it tells you the 3 parts to ATP production via aerobic respiration. So glycolysis, Krebs cycle, electron transport chain, 3 parts to this. So we're going to take a closer look here, and we're going to start out with glycolysis. Again, glycolysis, here's the essentials. This is what you need to know. We're starting out with glucose. That's our model molecule. We're going to end up with 2 pyruvates, or 2 pyruvic acid, if you will. So we're breaking a 6-carbon glucose down into 2 3-carbon pyruvates, essentially breaking down the glucose, breaking it in half. There are many metabolic steps that you don't have to memorize. What you should know, though, is that in the early steps, you need to sort of supercharge the glucose to make it reactive. So you're going to have to put 2 ATPs in. So for each glucose molecule, you're going to have to add 2 ATPs here and here. And in the end, you're going to get 4 ATPs out. So you're going to get 2 out here and 2 out here. And in the end, since we have to subtract the 2 that we had to put in, we're going to have a net gain of 2 ATP in this whole process. So these are the essentials that you need to know. Starting molecule, glucose, and molecule in glycolysis is pyruvate. We are putting 2 ATP in, and we're getting 4 out, which means that we have a net gain of 2 ATP in this process. You do not need oxygen for this process, and you can do this process easily in the cytoplasm. Every cell can do this. So glycolysis is a quick way of generating some ATP. Doesn't gain a whole lot, only 2 ATPs per glucose, and it does create a side product, a byproduct. If you don't deliver enough oxygen to your muscles when you're working out, then you're forcing your muscle cells into glycolysis as well. And that means that we're going to have a buildup of a lot of pyruvate, which in the case of human cells will be converted to lactic acid or lactate. Other cells have different types of metabolism. Yeast, for example, will then generate alcohol and do a fermentation process that generates alcohol. Anyway, so this is basically it for glycolysis. We're already ready to move on to the Krebs cycle. And the first intermediate in the Krebs cycle is citric acid, so therefore a lot of people also call it citric acid cycle. But a lot of people also give credit to the guy who figured it all out, and that was Mr. Krebs. So okay, so here we're starting out. We're having two pyruvate molecules coming from the cytoplasm, and now we need to enter the mitochondria all the way across both membranes, and we're gonna go into the interior, all the way into the interior of the mitochondria. In the process of going all the way into the interior, we are going to convert the pyruvic acid, into acetyl-CoA. That's very important. Acetyl-CoA. Acetyl-CoA has 2 carbons, so here's your carbon accounting, 2 carbons right here. Pyruvate had 3 carbons, that means we lost a carbon as CO2. Okay, so 1 carbon splits off as CO2, and now we're left with a 2-carbon compound known as acetyl-CoA. Acetyl-CoA will enter the Krebs cycle by binding to the final product of the previous round, which is oxaloacetate. Oxaloacetate plus acetyl-CoA. Oxaloacetate has 4 carbons. Acetyl-CoA has 2 carbons, and that makes the first intermediate in our Krebs cycle for the next round. That's citric acid or citrate, and it has 6 carbons. Okay. In the process of doing all of these reactions going around in the Krebs cycle, for every round of the Krebs cycle, we're taking a 6-carbon citrate and then gradually oxidizing the citrate to a bunch of intermediates until we end up with oxaloacetate. That, of course, has 4 carbons. And then that oxaloacetate will bind to the next acetyl-CoA, form 6-carbon citrate again, and we're going round and round and round. So what do we gain out of this whole process? So obviously, since the citric acid has 6 carbons and the oxaloacetate has 4 carbons, we're losing 2 carbons, and they're leaving us CO2. So we're going to lose 2 CO2 in the process here of oxidizing our citric acid molecule, or basically the acetyl-CoA that entered. Now, as we're oxidizing, the important thing really is that all the hydrogens that we are ripping off of the citric acid molecule here, when we are oxidizing the citrate, these hydrogens will be parked onto those coenzymes. Now they were NAD and then they become NADH. So we start off with NAD and then we make the NADH. And here we make some more NADH. So all of these hydrogens that are coming off of that, or the original glucose, if you will, they will be parked onto these coenzymes. And basically these coenzymes, they function as hydrogen taxis. The hydrogens will then be moved over to the electron transport chain. When you do that, then you have the big energy output. So the big deal thing about the Krebs cycle is that you are oxidizing the acetyl-CoA that entered the Krebs cycle completely. The carbons, 2 of them will leave as CO2. So 2 CO2 will leave in the Krebs cycle. And you're generating 1 ATP. Wow, we're not impressed with that, but that's not really the big picture or the big point here of the Krebs cycle. But generating or loading up all of these coenzymes with hydrogens, that's the big deal of the Krebs cycle. So we're going to generate a bunch of NADHs, NADPHs, FADH2s actually. So all of these hydrogen taxis are being charged up with hydrogens. And you can read through all of the steps. It's all explained here. It's kind of boring. But the big deal thing is our starting compound in the Krebs cycle is we're going to have acetyl-CoA. So let me back up. we're starting out here with the pyruvate that came from the cytoplasm. I'm going to move the pyruvate into the mitochondria. Along the way, we have a prep step, and that is converting the pyruvate into acetyl-CoA, which then enters the Krebs cycle, will be completely oxidized in the Krebs cycle. 2 CO2 will leave to then make a 4-carbon compound oxaloacetate, which then binds to the next acetyl-CoA. So the CO2, the carbon chains are now, the carbohydrate is completely oxidized, and we're gaining 1 ATP, which is not the big deal here, but the big deal is that we're gaining a bunch of NADHs and FADH2s. Okay, so that's the big picture of the Krebs cycle. And now we haven't generated a whole lot of ATP if you think about it. So for every glucose molecule, we generated 2 ATP net gain in the glycolysis, and then for each acetyl-CoA that entered the Krebs cycle, we're generating 1. So we generated 2 acetyl-CoA per glucose molecule. So we're gaining 2 total ATP in the Krebs cycle. So now we're up to 4, which is not super impressive. But here it comes. So now we're doing the electron transport chain. And the electron transport chain is basically a series of enzymes that are embedded in the inner mitochondrial membrane. And so all of these hydrogen taxis that were loaded up with hydrogen, the FADH2 and the NADHs, all of these hydrogens are now being delivered to the first enzyme here in this chain of enzymes that forms the electron transport chain. Now, a hydrogen, if you remember this from the chemistry lecture, hydrogen is the first element in the periodic chart. It only has 1 proton and 1 electron. We're basically unloading these hydrogens and splitting them into their subcomponents, which is a proton. That's the H plus here. These are all of the protons. And then the electrons, they will basically considered like the flow of electrons when you're plugging in an appliance. So you will be generating this flow of electrons going through the electron transport chain and powering these pumps. Now these pumps, what are they pumping? Well, they're pumping hydrogen against their concentration gradient. So they're taking these hydrogen pumps that are powered by the flux of electrons from these hydrogens. They will then use the proton, which is the other part of the hydrogen, and pump it into the space between the inner and outer mitochondrial membrane. So now you have a huge concentration gradient. So you're building up a strong concentration gradient. You're pumping all of these hydrogens into the space between these 2 membranes. So we're going to move them all over here. And this costs us some energy, and the energy is provided by the flux of electrons going through this electron transport chain. So now our energy is basically in a concentration gradient. We have generated a huge concentration gradient of a lot of hydrogen ions in space between these 2 membranes and virtually none in the interior of the mitochondria. And here it is. This molecule right here, the final step here, is called the ATP synthase molecule. This molecule will generate lots and lots of ATP as long as the hydrogen ions, these protons, are going down their concentration gradient through the pore of this molecule, the ATP synthase molecule. Picture it like the way you're generating energy when you have a waterfall, and you're letting the waterfall go over turbines that are then spinning, and they're generating electricity or some other form of energy. So basically you have a waterfall of hydrogen ions that wants to go down a concentration gradient, and it is allowed to do so through this ATP synthase molecule. And while we're turning here these turbines, we are recharging ADPs to make ATP. How much ATP? It varies a little bit from cell to cell and from book to book that you're reading. But for the purpose of this class, we're going with 32 ATP per original glucose, approximately. Okay, so the electron transport chain will churn out 32 ATPs for the equivalent of 1 glucose molecule. Okay, and this is the big payday. So we're getting paid now. We generated 2 ATP in glycolysis and 2 ATP in the Krebs cycle, and now we finally arriving at payday. We're generating an extra 32 ATP. Okay, so your book has sort of this energy balance sheet in summary right here, where we're saying, okay, so let's follow the ATP here, basically. If we just do glycolysis, we have this net gain of 2 ATP. Glycolysis is not the way to go, especially for a complex organism like the human body. So bacteria might get away with anaerobic metabolism, but that's about it. If you want to generate the energy that's necessary to run a big complex organism, you have to have a different way, and that would be aerobic metabolism. As soon as we have oxygen available and mitochondria, we can then run the Krebs cycle. Here was listed here, citric acid cycle, and then of course the electron transport chain. So in the citric acid cycle or Krebs cycle, we're generating another 2 ATP per glucose molecule. That's not too great, but really what we're paying attention to is the amount of NADHs and FADHs that we're generating. All of these hydrogens, they will then, the electrons from the hydrogens will power the electron transport chain, and the hydrogen ions, the protons, will form the basis of getting this concentration gradient going, and then we're going to release the energy that we have built up in that concentration gradient by letting the hydrogen ions go down. The ATP synthase molecule, think of it like a hydrogen waterfall, turning the turbines, and in this process we are generating lots and lots of ATP. As I said, the books are slightly different. So here this book is a little bit on the low end. So I always, well, here they're saying totals 30 to 32, with the electron transport chain being at 26 to 28. I have read books as high as 34 in the electron transport chain in under very ideal conditions. I always say, okay, let's settle for 32 here. And so the majority of books sort of under good conditions, you should be able to get 32 ATP out of the electron transport chain, plus the 2 from the Krebs cycle, plus the 2 from the glycolysis. 32 plus 2 plus 2 makes 36 ATP, and that's usually what I go with. Now, I would never give you a question on a test asking, is it going to be 34 or 36 or 38 total? So it's not going to be a question that splits hairs like this. We're going to go with 36 total. We're going to go with 2 from glycolysis, 2 from the Krebs cycle, and 32 with the electron transport chain, and that's under ideal conditions. Okay, so here we're just going to sort of go through this a couple more times to really enforce this whole concept of the aerobic respiration. Here is the part shown where we're converting the pyruvic acid that came from the glycolysis, the pyruvate right here. We're going to convert that. So this is a big old mitochondrion right here. We're going to convert the pyruvate into acetyl-CoA, and you can see that this has 3 carbons. Here we have 2 carbons, so 1 carbon is leaving as CO2. So that's here the conversion of pyruvate to acetyl-CoA. And here is another slide that gives you some information about how you can poison the whole system. And you find some very strong toxins in here that would kill you because they will stop the aerobic respiration system. And if you put a wrench into the system, you're just not going to be able to generate enough energy to run your cellular metabolism. Here's one, rotenone. That's it's like a pesticide that was used for some time. And it binds to the first component of the electron transport system, and then basically puts a wrench into the system and stops the flux of electrons. Then these pumps will not be powered, and you will not be able to build up your hydrogen ion concentration gradient. You probably have heard of cyanide being very toxic, or carbon monoxide. Basically, both of these molecules take the spot where the oxygen should work as the final electron acceptor and accept the electrons that have released their energy, plus the low energy hydrogen ions that come from that release their energy by going through the ATP synthase molecule, and so we're making H2O here, water, metabolic water. So, but if you're blocking this binding site where the oxygen is sort of waiting for these electrons and the hydrogen ions to combine to form the water, well, then you can't run your aerobic respiration either. And so cyanide and carbon monoxide, very toxic, because again, you will stop aerobic respiration. You will not be able to generate the ATP. And here's the final one. This one, oligomycin, it will plug up the ATP synthase molecules with the mouse here. It's just going to put a plug in there. And that one, of course, then if you don't release that hydrogen ion concentration gradient, well, you won't be generating those ATP either. Here's an interesting one, DNP, dinitrophenol. That stuff was used as a diet pill, of all things, for some time, with good success, because what it does, it makes the membrane here, the inner mitochondrial membrane, it makes it leaky to hydrogen ions. So now it's basically trying to, so if you have your water up there on the waterfall, you're ready to release the energy, and now the water found a different way around it, and it's just going downhill without going through the turbines here of, in this case, our ATP synthase molecule. So you're just releasing the energy from this hydrogen ion concentration gradient without going through the ATP synthase molecule. And it works because you're not generating a lot of ATP, so your cells are constantly burning more glucose or more food molecules to generate the ATP that you need. But people have actually died because this hydrogen ion concentration gradient does have some energy, and this energy will be released. And basically people have died from overheating, taking these diet pills. So now let's finish this topic here of aerobic respiration up. I know this this recording is getting a little bit long, but I have like 3 more slides and then we're done with this whole section here. So let's take a look at the ATP balance sheet, and I think everybody should be pretty clear by now. So we have a net gain of 2 ATP, and that's something that you really need to know. The net gain of 2 ATP from glycolysis. We have a net gain of 2 ATP in the Krebs cycle, and that's per glucose molecule that we put in. And then we have a net gain of, well, here in your book it says those 26 ATP. It really varies. It really depends on the cellular conditions. So we're going to go with 32 ATP from the electron transport chain. We also are gaining a bunch of these NADHs and the FADH2s, with the important part being these hydrogens that then build up a hydrogen ion concentration gradient powered by high energy electrons from these hydrogens. And so we're going to go with a total gain of 36 ATP per glucose. So this is sort of the theoretical yield. Depending on the cellular conditions, it could be varying a little bit. But anyway, for the purpose of this class, let's go with 36 total, 32 from the electron transport chain, or we call it also oxidative phosphorylation sometimes. So 32 ATP under really ideal conditions, plus 2, plus another 2 makes 36 total per original glucose. And again, depending on what book you read, you might go anywhere between 28 to even up to 38 total. So books vary on that. Now, here's a slide that shows you that it's not just glucose that you can metabolize for energy. Your body loves to use carbohydrates. It's pretty straightforward. You break down the complex carbohydrates into simple sugars, and so these monosaccharides that will go into glycolysis, forming pyruvic acid, acetyl-CoA, Krebs cycle, electron transport chain. So you go straight down here. You can also metabolize fats. Fats are triglycerides, dietary fats are triglycerides. The glycerol is a 3-carbon compound that can enter the pretty much here sort of at the tail end of glycolysis and then be converted to pyruvic acid and go down this way. The fats, the fatty acid parts from the fat, they have to be broken down into 2-carbon pieces. And while we have 3 fatty acid tails of 20 carbons each, so that tells you that each fatty acid tail can produce about 10 acetyl-CoA times 3, so 30 acetyl-CoA. That tells you how much energy is in fats. But anyway, so you can break down these fatty acid tails into 2 carbon pieces that will be converted to acetyl-CoA and enter then the Krebs cycle. You can even digest proteins if you like, although your body absolutely hates to do that for 2 reasons. Number 1, we need the amino acids to build up our own proteins. And also we have the amino end to deal with, which then generates ammonia, and it's sort of difficult for your body to take care of. So anyway, but it will be excreted with the urine, but your body really doesn't like to do this. But anyway, it's possible. You can break down amino acids into 2 carbon pieces, which then mostly enter here as acetyl-CoA into the Krebs cycle and the electron transport chain. So just to tell you that food in general, you can pretty much break it all down into acetyl-CoA ultimately that enters the Krebs cycle. Okay, and here's my final slide, promise, for the aerobic respiration lecture. So here's a summary slide that comes out of a general biology book. I just like it because it's summarizes everything that you need to know. And it shows ATP in like gold coins. So anyway, one more time for recap, and use this slide here to really, this is a really good summary slide. So anyway, so we're starting with glycolysis. That's our first step. Procedure happens in the cytoplasm, which is right here in blue. You can even write that down, cytoplasm. And we're taking glucose and we're breaking it down into 2 pyruvates, so 2 pyruvic acid. We have to put in 2 ATP dollars. We're going to get 4 ATP dollars out. That means we have our net gain of 2 ATP. Okay, we also gain 2 NADHs. Then we're going to move the pyruvates into the mitochondria. See this humongous mitochondrium right here, outer membrane, and then here the inner folded membrane. So 2 membranes. This is your mitochondrium right here. Okay, now we're moving the pyruvates into the inner space of the mitochondrium, and we're converting it from pyruvate into a 2-carbon compound known as acetyl-CoA. So for 2 pyruvates, we get 2 acetyl-CoA. We also get rid of 2 CO2 here in the process. The 2 acetyl-CoA will enter the Krebs cycle. For 2 rounds of the Krebs cycle, we're gaining 2 ATP. As we're doing 2 rounds of the Krebs cycle, we also get rid of the remaining 4 CO2. Remember, glucose has 6 carbons. 2 have left right here, 2 carbons as CO2, and then the remaining 4 carbons are leaving here now as 4 CO2s. So the glucose is completely oxidized to 6 CO2 in the process. Now there's nothing left of the energy of the original glucose, but the energy is now in these NADH2s, or the NADHs right here, and FADH2s, these here. All of these H's, there should be 12 hydrogens because original glucose has 12 hydrogens. So all 12 hydrogens are now parked onto these coenzymes, which then will deliver these hydrogens like little hydrogen taxis. They will bring them over to the electron transport chain, which is localized in the inner mitochondrial membrane, so in the inner membrane right here. The hydrogens will be split into electrons and hydrogen ions. The electrons, the flow of these electrons powers these pumps. The pumps are hydrogen pumps, which pump the hydrogen ions into the space between the inner and outer mitochondrial membrane. We will build up a high concentration of hydrogen ions in that space here, in this inner mitochondria, in its intermembrane space. And then the hydrogen ion, the concentration gradient, is now a form of energy. It's potential energy, and it will release its energy if you're allowing it to go down the ATP synthase molecule. And as these hydrogen ions going down their concentration gradient, releasing their energy, the ATP synthase molecule will build up a whole stack of ATPs. We said we're going to go with 32 total here under ideal conditions. But anyway, the point is, you see that here is payday. This is the big output. This is what we've been waiting for. Now, you might ask, hey, what's going to happen to the hydrogen ions and the electrons? Well, this is why we need to breathe oxygen. You need oxygen as your final electron acceptor. So the oxygen is waiting. It has to be waiting right here for these low energy electrons that come along and the hydrogen ions. Now, let me go back a couple slides because this is actually really important. You need to understand what's happening here at the end. It's been quite a few slides here, and this was kind of smallish, so but I do want to emphasize this, that at the end here, when the electrons are arriving, you need to have oxygen waiting, and this is really not very good either, so sorry. Well, it's kind of smallish here. So what's happening is you're forming metabolic water, we call that, because you're going to bind the electrons and the hydrogen ions. You're going to put them back together as hydrogens that then bond with oxygen to form H2O, which is water. Okay, so it's per oxygen molecule, so O2, that is, you need 4 electrons, 4 hydrogen ions to make 2 H2Os in this case. Okay, so I think we're done with this. I hope you're not getting too confused with this, but you should understand how your cells make the energy for all the metabolic steps that the body needs to do, and it's a lot. So here we have a kind of smallish here too with the oxygen. So, but you do need oxygen. Please don't forget this. You need oxygen as your final electron acceptor. So these electrons that have released their energy by going down this electron transport chain, and these hydrogen ions that have released their energy to form the to get all this ATP going. So now you need 4 H pluses. So let me write it down because it is really important. We need O2 plus 4 H pluses plus 4 E minuses, the electrons, and we're gonna form out of that, we're going to form 2 H2O, and that's why you need to breathe. So it is important to understand this. The four low energy hydrogen ions plus the 4 electrons, also low energy, they must bind to oxygen, and if you don't have oxygen, the whole thing will stop right here. If oxygen is not available, you're going to take the pyruvate, convert it to lactic acid. You're going to gain a grand total of 2 ATP, and that's it. And you cannot run your metabolism with that meager output of 2 ATP. So anyway, I am finally done with this here. I hope you can sort of sort through all of this, but use this slide here, use this summary slide as your guide as you're studying this. I realize this is not the easiest topic. There's also a worksheet that's posted in case if you're completely confused about this. You can work through the worksheet and sort of go through the steps a little bit slower than what we've been doing here. Anyway, so I'm done with aerobic respiration. We're just gonna then continue with transcription, translation, and replication next.